So today I'll be talking about some applications of targeted MS in biology and medicine. And all of the mass spectrometry techniques that I'll be discussing rely specifically on multiple reaction monitoring and using the targeted approach. So all of the global and shotgun techniques will not be discussed in the next 40 minutes. So what I'll cover today are the targeted hypothesis-driven MS experiments. Specifically, I'll go into an example of a cardiovascular study that was recently published by Hazmi Kashishian and our group. A phosphorylation study where we're looking at a recently funded grant called CPTAC2 in conjunction with target accelerator projects and targeting specific Phospho-Sites on certain peptides. Acetylation of Histones, a project that's led by Jake Jaffe in our group. And then some data analysis that if you decide to collaborate with the proteomics group, some things that you can do yourself with free software. So one question is, when would you use targeted quantification? And the answers are when you would have a candidate list of tens to hundreds of peptides or phosphopeptides that you want to repeatedly measure in a larger number of samples under varying conditions, different cell types. If you want to answer the question of what kind of relative quantification are you seeing change in quantification across samples, for example, in control versus stimulated cell lines, a lot of these topics have been brought up previously by the other speakers today and yesterday. Also in normal versus disease state or in tissue or plasma. And basically wherever a change of abundance or a calculation of abundance is needed. So we can look at exact peptides, we look at exact peptide sequences and we monitor for those to identify them and quantify for them. And we're able to observe small changes in abundance, smaller than the 2 to 5 folds that we normally say we can observe in shotgun type experiments. As a review, this is all what Hazmi had covered yesterday, but we use a technique called stable isotope dilution, multiple reaction monitoring mass spectrometry, in which we identify our target candidate proteins and look for specific, unique signature peptides that are unique only to the target protein of interest. And we have these synthesized in C13 and 15 stable isotopically labeled forms. And we use these as internal standards. So these are peptides, they're synthesized, and we receive them as dried powders. We can bring them up to almost any concentration, and we spike these into our samples, and they can be spiked in at different points along the workflow from digestion on forward. When these are spiked in a known amount, they're also present with the endogenous peptides of interest, and then we analyze them by nanoflow chromatography on a triple quadrupole mass spectrometer, again all explained by Hazmi yesterday. But the basic premise is we separate in time by chromatography, and then further we separate based on the peptide precursor, and then fragment it, and then monitor specific selected product ions. From here we get a trace showing intensity versus time, and here, shown in blue, is our isotopically labeled standard, and in red is our endogenous peptide that we're targeting. Because we know exactly how much of the internal standard we have, we can take the ratio of the light or red to heavy and blue, and we're able to observe precise ratios and give precise relative quantitation across samples in which the standards were spiked in. In this type of assay, we can quantify tens to hundreds of peptides in a single run. So there are some considerations when we go about wanting to target peptides of interest, specifically with relation to sample handling. So enrichment and fractionation in general improve our detection limits, even though they add a little sample handling complexity and perhaps the chance that some of the peptide itself may be lost. Overall, we're enriching for these target peptides. So the two areas of enrichment and fractionation are at the protein level. We generally use immunoaffinity, specifically of plasma, where we can deplete the top 14 most abundant plasma proteins, which get rid of about 98% of the bulk mass of plasma. We get rid of albumins, a lot of IgGs, and higher abundant proteins so that we can see the lesser abundant proteins to a larger extent. There's also potential to do cellular fractionation. If you're working in cell lines, we can go after certain subcellular portions with certain techniques. At the peptide level, we can enrich for phosphopeptides, as Philip had mentioned, using immobilized metal affinity chromatography. We can use anti-peptide antibodies or antibodies specific to certain modifications, like the ubiquitination gly-gly stub, or just general fractionation, like basic reverse phase or cation exchange. And again, what that does is it takes the one sample, separates it out into many different fractions, and then there's a better chance of actually seeing your peptide amongst the fewer peptides in any individual fraction. And further, these are analyzed by, again, nanoflow chromatography on the triple quad. And the idea here is that the internal standard will follow the analyte every step of the way and be processed identically all along. We also have the potential to use protein level standards when they're available, and those would be spiked in at the protein level fractionation step if that's used. So another important thing to note is that we're often limited by the amount of protein mass that we're able to load onto this nanoflow column step. So normally we're limited to about a microgram of protein, and we know that our lower limits of detection for these techniques are in the hundreds or low attomoles per microgram of protein. This is assuming no enrichment. If we do carry through the enrichment steps, because we're able to get rid of a lot of the protein mass, either through depletion or by splitting it up into separate fractions, we're still able to obtain around hundreds of attomoles, but now we're able to load a larger equivalent of protein mass at any given time on the column. And so here are just some examples with the case of plasma. If we wanted to analyze 1 microgram of neat plasma without doing depletion, just plasma as it comes right out of the body being separated, this equivalent volume for 1 microgram of neat plasma is 15 nanoliters. So we actually have to do a lot of dilution in order to get to this small volume on the column. If we're able to deplete, we can load a larger plasma equivalent volume, and we're able to get more on the column and just improve the detection limits. And so as you can see with each subsequent step, there's sort of an additive effect of the equivalent sample volume we can load, going from 15 nanoliters at hardly any sample separation down to about a milliliter if we use antibody enrichment. This is an example of a chromatogram that shows all the different targets that we're able to monitor in a given 1-hour assay. Each peak is just showing a different peptide, and the different shades within each peak are the different transitions that we monitor. You can see there's a lot of overlapping of the peaks, but the mass spectrometer doesn't really care about this because we tell it to look for certain portions of these peptides at given times, and it only looks for one at any given point before it moves on to look at the other one that's just adjacent to it or eluting at the same time. And because we're giving it such, almost like a horse with blinders, such fine little windows to look through, it really only observes each peptide one at a time. This is showing time from, the question was, is this an HPLC chromatogram? So it's showing the time of the peptides eluting from the column, but the actual measure of absorbance is from the mass spectrometer. So it's not a UV trace, it's a mass chromatogram, extracted ion chromatogram, and it's showing each peptide is a different color. And then the point is just we're able to detect a variety of peptides. So the key steps that we keep in mind when developing targeted quantitation assays are first the selection of the targets. Ideally, it's good to have empirical MS data available and observed preferentially by our lab because then we know where it comes from. But if not, data from the literature is equally as good. Something that's observed by a mass spectrometer once, we know we probably can see it again. Also, if it has been observed, we know that there's a good chance that we can have it synthesized into heavy and light target peptides, so we can characterize a quantitative assay. And by characterization of the targets, this was addressed by Hosmic yesterday, but we look at the targets and we go through and try to optimize the assay so that we get the most sensitivity for these specific targets. We look at the different transitions, the different gradients that can be employed to improve sensitivity. And then in the context of sensitivity, and as Mani had mentioned, we like to calculate the limits of detection and limits of quantitation. If we find that for some reason we did see the presence of a peptide in a discovery-like experiment, but when we go to build an assay, its LOD is actually quite high, it might not be worth pursuing if we can't get it to a certain level of biological relevance in the sample. Also, assay precision, if the peptide doesn't behave the same way chromatographically every time, it becomes a bit unpredictable, and then the results or the precision may be so poor that we can't get any useful information out of it. Once we go through these steps, we like to test on typical samples to demonstrate how the assay behaves in different cell lines and different plasma samples to show the reproducibility of the assay. So I will go over 3 different case studies that sort of touch upon the different areas of interest for our targeted assays. Case study 1 is a project that focuses on cardiovascular disease. And the biological model is the human model of myocardial injury, in which patients that are susceptible to heart attack are brought in and put through a planned myocardial infarction, in which on the operating table, there's thickening tissue in the heart, and the surgeons go in and try to thin that thickening tissue with alcohol ablation. And as they do this, this causes the same symptoms as a heart attack. So blood is measured from the peripheral plasma right here, and also in the coronary sinus very close to the heart. And the idea for measuring here is that any potential biomarkers that would signify myocardial infarction or heart attack early on could be directly measured without being diluted as they progress out into the peripheral plasma. And then blood is also collected from the peripheral plasma to show, even after dilution, if it's still able to be detected at several different time points post heart attack. And so this was a very large study that looked at a number of patients, and each patient had their own time course, so they acted as their own control. It went through a discovery phase where a list of candidate proteins were observed over the time course for each patient, and correlations between each patient were shown to carry it through to the qualification stage and prioritize the list of protein candidates, and further to the verification stage. So this discovery, qualification, and verification are sort of the 3 main steps we take the projects through from beginning to end to select candidates, make sure that they pass going from the peripheral body fluid to the body fluid that's further away, proximal body fluid to peripheral body fluid, and then to make sure that we can detect with a simpler, more targeted assay. So what's shown here are data from the discovery type experiments, in which case these were not eye-track samples. These were samples that were analyzed for a given patient. Each patient here is shown in a different color. And this first plot on the upper left-hand side is showing the percent normalized intensity, so 100 being the maximum, over 3 different time points. And you can see for this particular protein, the patients show a similar trend over those 3 time points. And that's also shown for these other 3 proteins. These are known cardiovascular disease biomarkers. So seeing this trend was very reassuring in that we knew that the samples were of good quality and showing good results. So the next 8 proteins shown down below are also showing very good correlation between the patients over the different time points. But these were candidate cardiovascular biomarkers, biomarkers that had not been fully demonstrated or tested for in the clinic. And the idea here is the physicians or the clinicians would be looking for something that would increase rapidly and potentially stay high, so that when a patient comes into the hospital, to the emergency room, an unknown period of time after a heart attack, these could be tested for, and if they were high, then they may be indicative of how recently the heart attack had happened. So the signature peptides that were generated for each of the proteins are shown in this table. This is not meant for you to read it. It's just meant to show the thoroughness, and we generate the light and heavy versions of each peptide. We look at lots of different characteristics. We classify the assays by the transitions. We always monitor a minimum of 3 transitions per peptide for selectivity and sensitivity. And then ultimately, the assays were characterized to calculate the LOD and the LOQ. So this is an example of some of the assay characterization that we do. Each different plot is a protein, and each protein has between 1 and 4 different peptides. And this is showing the peak area ratio, so that's the light peak area divided by the heavy peak area, in which the heavy peak area is our standard, and we always keep that fixed in all of our samples, and we vary the light amount of peptide over a concentration range. And the point here is just to show that it's linear, that each peptide may give a slightly different response. This is something Jake touched upon yesterday with his ionization efficiency. And let's see, let's zoom in a little bit. So AEBP1 shows 4 different peptide sequences at the top. The steeper the slope generally tends to indicate that peptide has a stronger signal, may be more sensitive. We may be able to detect it a little bit better in the assay based on just the peptide itself, but this may not give us any indication about how well the peptide digests from the protein. But again, this is just some of the preliminary characterization that we do for every targeted assay. So in the context of this study, there were 6 different patients shown on the y-axis that were monitored, and each of the different colors on these plots represent the different peptides from the proteins, and the proteins are shown at the top. So if we go down a row, you can see the correlation or the trend over time for each of the patients tends to be very good, with ACLP1 showing a downward trend, some proteins showing a little bit up before tailing off, some proteins showing a gradual increase in the trends. So when the clinicians see this, this is something that's very promising. Also, I want to point out that the concentration amount here is shown in nanograms per milliliter of plasma. So this is the levels at which we're able to detect and the precision with which we're able to detect. Each point represents the median or the mean of a process replicate, with the error bars representing a standard deviation. So you can imagine if you were a clinician, you might want to pick time points or create a panel of these assays so that when a patient comes into the hospital, you can say, oh, if ACLP1 is low and TMP1 is high, it could mean that the person had a heart attack. And if you can correlate to the time point, you might be able to narrow down when that actually happened. Okay, so any questions on that study? All right, and Hazmik Kashishian will be the one to answer detailed questions about that, and there's a paper reference at the back too. So case study 2 is addressing quantification of phosphopeptides. As Philip presented earlier, phosphopeptides are very important with signaling pathways. You can see many examples here. I'm gonna divulge, I'm not a biologist, I'm an analytical chemist, so what they do isn't as important to me as can we detect them and how well can we detect them. So on to the next slide. We've been undergoing a study in our group where Philip has been arduously going through IMAC enrichments and detection of peptides in discovery phase projects from breast cancer tissues. And what he's been able to show are that many of these peptides and specific Phospho-Sites have been detected out of the discovery data. So this is very promising to us. Again, as I mentioned earlier, we like to see empirical MS data for peptides of interest, because then we know we have a pretty good chance of observing them again in a targeted mode. So anything with a little checkbox indicates that Phospho-Site indicated in the second column was observed for a particular peptide sequence. So using this list as our sort of initial set of candidates, we designed those peptides in synthetic forms with isotopically labeled amino acids. And this is the general workflow that we use. Not only are the phosphopeptides of interest, but also they're non-phosphorylated forms. So we can say, we can get to that question about stoichiometry that Philip had mentioned earlier. So in general, if we're going through an IMAC enrichment, we like to start with a larger number of cells. We're actually working on titrating this down right now. So initially, this is many, many cells. We're hoping to drop it to a half or even less than that. But this workflow can be done on cell lysate or a tissue lysate, or even plasma if necessary. We take it through a trypsin digestion step, so we get a bunch of peptides, and then at this point we can add our labeled standards. If we are monitoring simply for the non-phosphorylated peptide sequences, we can add the peptides and analyze right away. If we're looking to monitor for the phosphorylated peptide forms, then we add those peptides, carry it through the IMAC enrichment, and then analyze on the mass spectrometer. And so this is just an example of the two proteins, YAP1 and MAPK1, and a particular Phospho-Site. So the phosphorylated form of each peptide is shown on the bottom, with the blue being the internal standard and the red being the endogenous level that we're able to detect. And on the upper plots are those same peptides in their non-phosphorylated form, so the same sequence, but minus the phospho group. And we're able to detect a tiny bit of the YAP1 peptide and a little bit more of the MAP kinase 1. So again, this is preliminary phase in our group. Phosphopeptides, I think Hosmic had mentioned yesterday, there are general rules we like to follow when we're picking peptides, signature peptides from proteins. And obviously, you can't be so choosy with phosphopeptides, because if the specific site is of interest to you, then it may have some amino acid residues in that sequence that are ones that we'd try to avoid. But in general, if you can't avoid it because they're in the sequence, we generally like to target the sequence anyway. So right now, we've got an assay targeting 31 different proteins with almost 90 total peptides, 47 of them in phosphoforms and 42 in their non-phosphorylated forms. So some of these peptides have multiple Phospho-Sites. Any questions so far? Okay, the other thing I wanted to point out is when we use the IMAC enrichment step, the equivalent input amount that we use is generally a milligram of protein, where that is about what we load on the column. Whereas if we're not doing the enrichment step, we wind up analyzing about 1,000-fold less than that, because we're not enriching and we have to analyze directly. So while these amounts can't be compared apples to apples, I can show you an example of how we do compare them. So again, these peptides don't behave exactly the same way as our normal peptides that we pick for targeted assays, but we do try to characterize them and make sure that they behave well enough if we do have choices. So this is an example of their HPLC retention characteristics, where this is their measured retention time on the y-axis, and this is just a hydrophobicity calculation on the x-axis. It just looks at the amino acid composition only and calculates a hydrophobicity score. And so it shows that there's a good correlation between the HPLC method that we use and their hydrophobicity score. This also helps us when we have so many peptides to target. We can sort of be able to predict where they should elute chromatographically, and they should elute chromatographically in the same place every time. So on the bottom plots, this just shows examples of, for example, a peptide sequence. The star represents the heavy amino acid that we have incorporated with C13 and N15. And then the little red ball is the Phospho-Site. And this is showing an extracted ion chromatogram for up to 5 transitions that we monitor. So for the same peptide without the phospho group, we would monitor a similar number of transitions, but those transitions would be different because it would have it wouldn't have the phospho group on it anymore. And also an interesting case where we have peptides with the same exact sequence but 2 different potential Phospho-Sites. If they're occupied not close together on the peptide sequence, we can usually resolve them chromatographically. So they can separate in time, which for us makes things a little bit easier, but we do have cases where we have 2 Phospho-Sites that are only 2 amino acids away from one another, in which case they don't resolve, and I'll show that in a minute. So again, back to peptide selection rules. Normally, we like to pick at least 3 transitions per peptide, but with phosphopeptides, especially in cases where you have several potential Phospho-Sites on a peptide, it's really important to figure out which Phospho-Site is occupied. So what we'd like to do is pick fragment ions that will help us discriminate. Here's an example of this peptide. And this is an example of Skyline showing the extracted ion chromatogram and all the transitions that we're monitoring. And you can clearly see two well-resolved peaks from one another, but we have three forms of this peptide we're trying to separate. We've got this one, and these are all standards, so we know how much we're spiking in, where this serine is modified. And then we've got these other two forms where the two N-terminal serines are modified, but they're so close together in the amino acid sequence that we actually can't resolve them chromatographically. So they appear together in this one peak. And the only one ion that we would be able to use well enough to differentiate between the two is the Y8 ion. So that falls right between those two serines. So not only do these peptides have to fragment, but they have to fragment to produce the Y8 ion. And you may not be able to see it from here, but the Y8, let's see, it's 857 mass to charge up here, and the Y8 down here is 937. So we would be able to figure out, based on that one fragment ion, which peptide's which. But all of their other transitions are, in fact, the same. So these are some of the challenges we see when looking at differentially phosphorylated peptides. Are there any questions? It's tricky. Okay. So one question might be, is IMAC enrichment necessary? And unfortunately, I say unfortunately because it's not a trivial thing, it is necessary. Some of these peptides are so low abundant in general, and then the actual occupancy of that Phospho-Site is even less than that. So this is just showing an example. If we take a sample, lyse the cells, and load 1 microgram of that sample on the column, we really don't see any appreciable signal for this one peptide. But if we enrich the sample, we take a milligram, carry it through that IMAC enrichment process, and load the equivalent of 300 micrograms on the column, then we're able to see a much stronger signal. So for many of our applications, unfortunately, IMAC enrichment is necessary. But the good thing is that we can spike these standards in before the enrichment, and we can make sure that sort of the sample handling variation that may occur is taken into account. So part of what I'm showing you today is not just the applications of what we're doing, but some of the nuances of how we analyze the data. And again, these are some features in this program called Skyline that is able to read in the raw data and visually represent to us easy little snapshots of how the data looks and quickly go through large numbers of data. So just to refresh your memory again, this is looking at an extracted ion chromatogram for a particular peptide where the blue is the internal standard and the red is the light or endogenous peptide coming from the sample. If we look at just that red signal, and now we want to look at the pieces that make up that red signal, it's made up of 5 different transitions shown here in different colors. So each of those transitions needs to co-elute. If anything comes a little to the right or a little to the left, we know that it can't be arising from that peptide because that peptide has to fragment the same way every time. It reaches an apex and then it tapers off. So another thing that's useful for us is we have this plot that shows the contributions of all of the different transitions normalized to 100% across all of the samples. And this bar chart on this bar on the left-hand side is based on our spectral library. So we have a reference spectral library that was observed early on with the standard peptide. And you can see that it's very, very similar to the analyses that we're looking at across all of our other samples. So this is just a quick sanity check to make sure, does the spectral library represent what we're seeing in our samples? Yes or no? If it does, great. If it doesn't, it may be the wrong peptide. Okay, so using this type of approach, we're able to address the question of how does a phosphorylation site change in different cell lines. So here we've got 3 different cell lines, and one with many, many replicates, and the other 2 with 2. And we're looking at 2 different Phospho-Sites in June. And we're not expecting the Phospho-Sites to have the same abundance or even the same relative abundance in the samples. So what you can see is the one cell line has some kind of base phosphorylation level, and it goes up a little bit in the next cell line for site S63, and even higher in the 3rd cell line. But you don't observe that same trend for the other Phospho-Site. So this gives us a little bit of confidence that we know we're not seeing effects due to sample handling. If that was the case, we might see the same trend for both peptides, but instead we're seeing differential phosphorylation at these two sites. What's shown on the left-hand side is peak area ratio. So this is the ratio of the light peptide to the heavy. So we're seeing this in terms of a fraction, essentially. We can multiply this by how much heavy we added in. It's 100 femtomoles in these samples. So we're looking at 1.5 femtomoles here, 2.5 femtomoles here, so low amounts, but still within our limits of detection. And so to answer the question about stoichiometry, this is, again, we're sort of learning as we go a little bit and making sure before we give straight out answers that everything makes sense. So this is showing, again, those two examples from June. And the blue trace is showing the peak area ratio for the 3 different cell lines of the phosphorylated peptide form based on 1 milligram of protein input. And the red is showing the unphosphorylated form based on 1 microgram of protein input. So even though it looks like there's a lot, like on this plot, it looks like there's a lot more of the phosphopeptide. In fact, there's not, because we put in 1,000-fold more protein input. But you can see the change in the non-phosphorylated peptide is very small, but the change in the phosphorylated peptide is larger. So these are the kinds of things that we're looking at now to get at answers of what are the changes in the Phospho-Site stoichiometry versus how much of the protein abundance is changing in the sample. So the third case study is a project that is led by Jake Jaffe in our group. And again, he can give you a lot of the more interesting and biological details more than I could. But it's targeting histones and their different post-translational modifications. As you know, histones are very important for transcription, and they've got quite a variety of post-translational modifications. And for the mass spectrometrists, they can be quite challenging because many of these modifications are very close in mass. Because we measure mass, that can make it quite challenging. So histones in general are not amenable to proteomics approaches. We've heard the past day and a half that trypsin is one of the enzymes that we use practically religiously because it cuts very specifically at lysines and arginines. But if you look at the sequence here, all the lysines and arginines are shown in red. And in general, we don't like to target peptides that are smaller than 6 amino acids long. So you can see this whole stretch of this histone H3, we wouldn't be able to detect any of these things because many of the peptides are too small. So the workflow that's done in the group is that the histones are propionylated, which goes after the lysines. So each of the lysines now has is now modified. Trypsin doesn't recognize it as a normal lysine, and it won't cleave there anymore. So now it cleaves after every arginine, and it generates these different peptides that are larger and more easy to detect in the mass spectrometer. Also, as it points out, it helps with retention. So propionylating the lysines makes the peptides a little more hydrophobic, and so they're able to bind to the columns, and we're able to see them better. Now, because of the very near mass modifications that we're seeing between acetyl and methyl 3 groups, for example, on this one peptide, this is the same exact peptide sequence, and the mass between these two peptides, because of this different modification, is very, very small. This isn't something that a triple quadruple mass spectrometer is designed to handle very well. So it has a difficult time differentiating between the two peptides. So an example on the top is showing the extracted ion chromatogram for the transitions that could satisfy either of these peptides, but this is the correct peak for this first peptide. On the bottom is an extracted ion chromatogram from a different mass spectrometer. This is a Q-exactive. And the main difference here is that it's a high resolving power instrument. So not only can you see high resolution in the MS, but also for the fragment ions, in which case you can see changes out to such small decimal places. And so in this case, when the data is analyzed by Skyline without really any manual intervention, the correct peak is identified for this version of the peptide based on the data. However, for this version of the peptide, when analyzed on a triple quad, again, this is the same data file, Skyline incorrectly identifies a peak that is not the right one. So it's going after the same peak and calling these two peptides essentially the same thing. So this is not correct. Over here on the Q-exactive, because you're able to monitor for many, many more transitions at any given time, because we look at all of them every single time, it's able to point out that the correct peak, where most of the transitions are overlapping, is this small one in the lower right-hand corner. And you can see these sections in green are blown up 20-fold. So you can see the detail that's down very, very low. So this is one of the reasons this project uses Q-exactives, not only because the small mass to charge, but also the more plentiful information that the Q-exactive gives you. So just to illustrate this point a little further, this is what the data would be read in from Skyline. It would just really be only seeing a few transitions for a peptide, or it would be seeing everything from a given peptide. And really, we call this post-acquisition data processing is the strength here, because instead of monitoring only the 3 or 4 or 5 transitions that a triple quad would be able to give you, it can monitor as many as the mass spectrometer can give you on a Q-exactive. And the Q-exactive gives you as many as it can every single time, because it scans the entire range. Whereas on the triple quad, it really just scans little tiny windows around where you've already told it to scan. So main difference between those two. So back to the histone workflow, they use a SILAC approach in using heavy arginine and not heavy lysine. And what this does is this generates a standard that can be it generates a standard that can be used to analyze against any other cell lines of interest. And the modifications are always in the biologically relevant range because they came from a cell line. Nothing additional was spiked into that standard. Or the other option is synthetic peptides, where we're using the same approach that Hosmic had mentioned yesterday and that was used in the cardiovascular and the phosphopeptide workflow. These peptides are synthesized, spiked into the sample in known amount, and then you're able to get a quantitative analysis rather than relative changes across samples. Also, because you're spiking in specific peptides and not a whole lysate into a sample, it winds up making the sample a little bit less complex, but you're only able to target the specific peptides for which you have standards, whereas in the SILAC, you're able to monitor anything that might be present in that SILAC-labeled sample. And this shows the workflow. So Jake and Jordan Taylor and others in the group have worked on making this very industrialized and as automated as it can be in our hands. But this shows the workflow. It lasts a couple of days, and after a few days, they wind up with a 60-minute assay or a 90-minute assay, depending on what they're targeting. Let's see. Oops, sorry, and this just shows if it's the SILAC mix, which would be the 90-minute assay, looking at as many modifications as possible for histones, or if the peptide standards are used, they're spiked in right at the end, and then the 60-minute assay targeting just the H3s is used. And this is the summary of the assay. So again, it's using the Q-exactive instrument, which is a smaller benchtop instrument, but very powerful. These show the column dimensions. It's using a targeted method. So even though this is an instrument that's used for discovery proteomics, it can also be dual-purposed for targeted proteomics as well, so specific targeted projects. When using the SILAC approach, all histone peptides can be monitored over a 90-minute assay. And when spiking in just the synthetic peptides, again, it's a 60-minute assay monitoring for H3 only. And then data analysis is through Skyline. And in the end, what Jake winds up getting is a lot of data that can be analyzed through clustering. So he looked at a large variety of cell lines, and looking at the different modifications can tease apart what modifications may be of interest for certain phenotypes, for certain cell lines. So the last section of this talk is on data processing and analysis. Skyline is a software tool that started at the University of Washington in Mike McCoss's group. It's actually developed by a former Microsoft employee who now doesn't mind living in the academic world. And it was generated, it's actually, they call it a document editor. So when you first open it up, it's hard to know what to do with it, but overall it generates targeted MS methods for triple quads. It also is able to work for the Q exactives and the higher resolution instruments as well now. So these are some improvements that were made recently. It aids in method development, it processes raw data files, and it can do some basic calculations for you. So if you have a limited data set and you don't want to go lock your statistician in a room somewhere, then some of these things you can do yourself. Also, it generates reports that are directly output that we can use with Quasar. And Quasar Monty got into, but I'll just show some use case examples. So Skyline, again, it's this document editor, and this just shows a little example of how we sometimes use it. We can use data-dependent search, shotgun experiments, and create spectral libraries within Skyline. And then from those spectral libraries, we can use those to prioritize and pick unique peptides. When we do that, we can generate a targeted peptide list, and the output from that is a vendor-specific triple quad method, or a method for any other mass spectrometer. Fortunately, the way Skyline was made, it can be used with any of the different vendor instruments. It's free, so there are a lot of benefits to it. After the data are acquired, you bring the data in, and it automatically picks the peaks and integrates them. And then you can go through manually, and years ago, we did go through manually each and every of the peptides and each and every of the samples, but now we've got some shortcuts. And in the end, you can have comprehensive result reports that when output can be analyzed in Excel, in R scripts, or in Quasar, which is what we do. So this is an example of what Skyline looks like when you have it populated. So on the left-hand side are all of the peptides that we're monitoring. They're broken down into their different proteins. So we like to monitor for a few peptides per protein if they're available. On the top is showing a spectral library. So this is an MS spectrum that was observed perhaps in a discovery experiment or by infusion of the standard. And what we can do is make sure that when we see a peptide coming out of the HPLC chromatographically, that the transitions, the 3 transitions that we're monitoring, agree with the 3 transitions that are observed in the spectral library. So there are a lot of different calculations. There are rankings, dot products to make sure that the identification, because we're just looking at a small subset of these transitions, agree with the spectral library to help make sure that our identification step is correct. Okay, now for improving the data analysis, you can imagine when we run calibration curves, we tend to do everything in triplicate or quadruplicate, and we have 8 to 10 point calibration curves. So this quickly expands to a large number of concentration points. What this shows is a calibration curve where this is our blank. So you can see the blue is the more intense signal, and that's kept fixed over all of our different concentration points as the red increases. And if we look at it in this view, we can sort of quickly, by peptide, we only have one peptide selected, we can quickly look and say, oh, that looks right, that makes sense. The red keeps increasing. This plot is showing our peak area view, in which case our heavy peptide is held at a fixed concentration in all of our samples, and that's constant across all of our samples, so we know that probably the correct peak was picked and everything looks good. And the red is increasing in groups of 4 because we did everything in quadruplicate here. So it's just a sort of a quick way of viewing the data. This upper plot is retention time, which doesn't look so pretty in this view, but the scale is not very big. So there is a little variation from run to run, but this allows you to observe and see if you have any constant trends. Like if your retention times are shifting up in one direction, then there might be something wrong with your HPLC. So it gives a little bit of sanity check to your data. So once everything passes out of Skyline, we can export it in a specific report format and bring it into Quasar. So Monty went over a lot of the specifics like calibration curves, reproducibility, LOD, but I just wanted to show some examples of how we use it. So again, I don't like having to go through each individual sample because 90% of the time, Skyline does a really good job of picking the peaks. So I want to trust the software, especially when we get into data files where we've got hundreds and hundreds of peptides and hundreds of samples. So what I like to do is look at Quasar and say, okay, this is my linear response curve. This is my logarithmic response curve, just fit on the logarithm, or sorry, just shown on the logarithmic scale, not fit. But I want to see, are there any outliers or anything that just doesn't look right? So in Quasar, we have these little flags, and they'll be indicative of the potential that an interference is being detected or that the CV of your replicates is too high. So for this green case, you can see that one little green triangle, it's a little bit higher than the rest. So what I might do is I might identify which concentration that is, which peptide it is, which transition it is, which is shown in this table. I'll go back to Skyline and just look at that set of replicates and identify maybe it was an incorrect integration, maybe something went wrong with that injection. And so it could be something that's fixable in the data rather than having to go and reanalyze the sample. And here's the same case here. This is probably more indicative of something going wrong with the injection because all three transitions are affected, whereas the others group nice and tightly, much higher. So I would probably go back and either the wrong peak was picked by Skyline or something might have gotten wrong with that one injection. Chances are it might not be the injection because these data points shown here and here are all coming from the same sample series. So the samples that were injected here for this peptide, it's the same sample here analyzed all at the same time. So the chances of this being a wrong injection, maybe it is, but either way, it just pinpoints the problematic things I need to go look at. Also, we can get an LOD distribution from the response curve. So this is using the calculations Mani explained. And as I mentioned, we normally see the LODs in the low hundreds of attomole. So this is showing the median is at 200 attomoles per microliter. And this is showing the distribution of the LODs across all the peptides. For this example, there were 125 peptides in this assay. And again, the CV shown per concentration. This is just a categorical scale. It's not meant to be fit, but you can see that the CVs decrease as you go to higher concentrations, which is what we would expect. And as long as we don't see any CV hopping up higher, then we can have confidence that these data were processed pretty well. So one example of data that we were working with a collaborator on, we were looking at just a simple 4-point response curve with the blank. The blank we expect to have high CVs because we're not actually measuring an analyte in the blank. But you see the CVs drop down as the concentration increases in this one response curve. But the new response curve we received before I actually started doing a lot of analysis in Skyline, I just imported the data, quickly processed it, exported it to Quasar, and I got this plot, and I said, oh my gosh, what the heck happened? Did I do something wrong? And then I realized, if the CV is popping up and then down and then up as the concentration increases, there's something wrong with these two sample sets. So I called our collaborator and I said, hey, I'm just curious. When you did your sample dilutions, did you do it in 2 different series, and did you start with the 10 to dilute to 1 and start with a 5 to dilute to 0.5? And he's like, yeah, how'd you know? I'm like, you messed up your dilution, and I can tell really easily. So there was just 1 point out of the triplicate series that was off, and it was off for both the 10 and the 1, and that caused those CVs to be driven up, and it was quickly identifiable here with Quasar. So in summary, I hope that I've helped explain that targeted MS is an effective approach for quantifying peptides, especially for hypothesis-driven experiments. Post-translational modifications like phosphorylation, like acetylation and methylation, they pose challenges, but they're far from impossible. We have a lot of tools in our bag, and there are more being developed as we are challenged to make things a little bit easier for us. Also, we're able to monitor tens to hundreds of peptides in a single analysis. I think, what's the lab record? 240 peptides? 400? 400. So the lab record right now is 400 peptides that can be targeted in a single LCMS run. So we're just creeping up and sort of pushing the envelope with what the technology will allow us to do. And then finally, Skyline and Quasar software, I'm big fans of both because, like I said, I don't like to have to look at each individual thing if it can be avoided. I like to trust the software, and these help provide some checks and balances to make sure that the data is of the quality that we need it to be. Okay, so here's some references. This is the reference to Skyline, and again, that's online. The web link is in the handout, and this is just showing some examples of the cardiovascular work and a review at the end. All right, any questions? Yes?
Can you monitor glycosylation, copying glycosylation in terms of what residues are glycosylated and what type of sugars are there?
Okay, I only heard glycosylation and what kind of sugars are on there. So
Can you, like you talked about, and actually the previous talks about protein phosphorylation and the glycosylation and acylation. And what about glycosylation? Did you ever have a project that detected this?
Okay, so that's not, as far as I know, a project that has been done in our lab. Glycosylation, I think, can be tricky because depending on the structure of the sugar side chain, that in itself can fragment off. So it may be difficult to identify the exact form. To my knowledge, I don't believe there are any labs outside of pharmaceutical companies who have a drug product with glycosites that specifically have targeted assays for that. But ideally, if a standard could be made that's isotopically labeled, that would behave the same way as the analyte of interest, whether it's glycosylated, whatever form the sugar chain would have, then they should behave identically, and we should be able to characterize it.
And another question, you showed this study for myocardial infarction of the peptides, right? So you follow the increase in different protein biomarkers. So what was the fold increase for these proteins?
Oh, so the question was, in the cardiovascular study, over the different time points for patients, what was the fold increase?
Yeah, so maybe these are just fluctuations, or are these real increases?
Well, okay, so the fold increases were first observed in discovery data, and those were statistically calculated to make sure that they were not an effect of protein load or sample handling. They were real changes in those specific proteins, and that's how they got down further to the triple quad assay lists. In the triple quad assay, then the peptides from those particular biomarkers were monitored more vigilantly, and the fold changes that were, like, I can't tell you specifically the fold changes because there are a lot of proteins and a lot of peptides. But the manuscript, I think, has a lot more details about the specifics of the different fold changes and how they were differentiated from just global changes in the proteome, if that's what you're getting at.
The reason I'm asking, I was involved in some study that tried to detect new biomarkers for cardiovascular diseases, like something similar. So we actually studied the change in the activity of proteins, because many of these biomarkers, the concentration doesn't change, but the activity can change enormously. In order of magnitudes. And it was shown that the activity is actually more reliable biomarker than the actual increase in proteins, which fluctuate.
Yes, yes. Yep, so we didn't monitor for activity. It was just...
You did not correlate for the activity only the levels?
No, I don't believe. So the question was, in that paper, was activity of those proteins correlated with the abundance of the proteins, the change in abundance? No.
So, I mean,